So, RAD sequencing data, as we've been talking about all day, has certain characteristic features, and there are a few different software packages out there for dealing with it. Just to give a little bit of history, like Mike said, he and I were both at the University of Oregon together when the RAD protocol was being developed. Mike was a central player in actually coming up with the molecular biology part, and I was just there tagging along trying to analyze the data. Mike and I and Julian Ketchen, the three of us, were all there independently writing code to handle RAD data. So Mike came up with his own stuff. I came up with my own stuff. Julian Ketchen, who's now faculty at the University of Illinois, had worked for Intel for a couple of years before he went to graduate school. So you can guess which of the three of us came up with the most user-friendly, best performing computer program. So the end of the story is that basically a lot of the stuff that I wrote, the algorithms and code and stuff, got folded into those stacks computer package. So there are some other programs out there. So PyRad, AfterRad, what am I missing? Rainbow, I think. There's a couple other things out there for handling RAD data, all with kind of the same idea of what they do. PyRad, for instance, is designed or is intended for phylogenetic stuff where the sequences are a little more divergent. And I haven't really played around with those other programs, so I can't really talk about relative efficiencies and performance and stuff.
I know for AfterRad, our postdoc that developed it, he doesn't have time to work on it anymore, so I would actually, slightly coming from my lab, I'd say stay away from it because it's not being used.
Yeah, so we're bringing endorsement there. So actually that's one thing I was going to say about Stacks, which is that it has a really active user group. I don't know, how many of you all are on the Stacks list, the email list? So you probably treat it like spam because you get so many messages with people asking questions, but people answering questions also. And Julian is on there like almost every day answering people's questions. So it's a really remarkable sort of resource that Stacks has that can help you work through this. So that's really nice. Okay, so I just want to talk in sort of in general. Here's just kind of a rough idea of the different things you could do with RAD data. So for instance, you might start with raw sequence data. You go through a couple steps on clean and demultiplex. Because depending on if you have a reference genome or not, you might align your reads at that point, or you might just identify loci de novo. You can call it genotypes and then do whatever it is that you want to do down at the bottom. And so this is basically like a different version of what's in the handout. You can imagine different paths through this sort of workflow where you're combining different steps. And this is where Stacks sort of gets its model, is that Stacks has all these different modules that you can combine in different ways to get different sort of workflow depending on what your situation is. One thing I'll just mention, Stacks, we've been using the Unix command line. I think you all are sold on the idea of Unix, but just to point out, it's been around for a long time. Let's say 1969. And for Mac users, this is one of the reasons why Macs are nice, because OS X is built on Unix, so you're already working in the Unix environment. And again, hopefully most of you all are familiar with a lot of these basic Unix commands. A lot of the abbreviations can be, they're all made to be as few letters as possible in the Unix commands. And some of them, the abbreviations are obvious. Some of them are not so obvious. So how many of you all have used Unix before today? Well, most of you. So you're all mostly pretty familiar with it. So, like I said, Stacks is a set of modular programs. You can run them independently. You can pick and choose what you want. Or there are sort of these wrapper programs that are part of stacks that will string together a number of the modules into sort of a commonly used pipeline. I bring up Unix because, so Stacks has three different types of interface, sort of a user interface. The first is command line with text files for inputs and outputs. So this is all based on Unix. This is essentially what I use almost exclusively. And I think it's probably what most people use who are doing really hardcore Stacks analysis. They're just doing everything on the command line, text files and stuff like that. It also has a MySQL database, which is basically a way of dealing with your data and making it good for doing all kinds of different queries of your data, spitting out filtered things, you know, if you want this set of individuals that at loci that are genotyped in X number of individuals with this minor allele frequency and this minimum coverage or whatever. You can do all sorts of searches that way. There's also a web-based interface, which can be maybe useful for visualization and stuff, but I know at least on our cluster at the University of Idaho, these two things are problematic, and they're not even on the Stacks version on the cluster. Does anyone use the web interface? A couple of you. So it may or may not be useful. Basically, you can get all the functionality with the Unix command line sort of. Okay, so like I said, there are a bunch of these modules. These are kind of the main ones. So process rad tags, which does multiplexing and quality filtering. Clone filter to remove PCR duplicates from paired end data. Stacks, which is the de novo identification of loci. This runs per individual. So Stacks is run separately on each individual sample, and it identifies the set of loci and calls genotypes per individual. I guess one thing to mention, so Stacks uses a maximum likelihood algorithm to call genotypes. It does not use a Bayesian thing with allele frequency information or things like that. So it uses a maximum likelihood algorithm per individual to call genotypes. Talk about that later. So this is if you don't have a reference genome. If you do have a reference genome, there's this program Stacks, which calls genotypes based on alignment files. So alignment to a reference genome. And Stacks makes a catalog of loci. So basically, a catalog is just the list of all the loci in the set of samples you have. And then individuals are matched against that catalog. So that now you know which locus is the same across individuals. And then there are a couple of ways to output data. So you can output genotype data and use it in downstream other software, and this will output data in all sorts of different formats. Or you can do a bunch of population genetic statistics within this program. It's called populations. Okay, so like I said, this was from the Stacks paper, just showing how you can kind of make a big workflow. And then this is what I put in your handout. So this is essentially the same as what you saw before. So in the data that you have, did anyone get the full big tarball?
No.
Or does everyone have the small tarball version?
So just to let everyone know, the big ones is available, so if later you want to download it, it's there on that link, so you just have to download it.
Okay. So these blue boxes are the data files, types of data files that are in that tarball. The difference between the big one and the little one is that, well, first of all, I should just say these data sets are massive. So I had already subsetted down from the actual study, and I didn't subset enough. So then I subsetted even more. So I gave you just sort of samples of FASTQ files. They come off the sequencer. FASTQ files for each sample. The big one had 2 populations with almost 20 individuals in each. The small version, I think, just has 1. Then here are SAM files after being aligned to the reference genome. Again, I think the small tarball just had 1. The big one had all the individuals from the 2 populations. And then stacks output files down here. So I just want to sort of walk through this a little bit. So these data were RAD data using Mike's protocol, which means, as he said, the cut site can be on either read in any pair. And so the first step is to flip them. So there are a few bits of code probably out there floating around to flip these reads. I included one in here that you can try out. Basically, it just looks for the presence of a correct barcode and cut site and either flips it or doesn't flip it, or tosses it out if it doesn't find one on either side. Then process rad tags goes through and looks for the cut site and a correct barcode and demultiplexes the data into a file, or actually a pair of files for each individual sample. So you have an example of that output.
Oh, sorry. Did you mean by flip repairs it gets it in the correct, like, 5 or 3' orientation? Or, like, from the restriction enzyme
Yeah.
site to the
So it, I'll show you an example in just a bit. It basically, the output of this, so the raw FASTQ files you get off the sequencer will usually have like an R1 file and an R2 file, and they're lined up in pairs. But the cut site might be either an R1 or an R2. The output from this split code puts the cut site ends all in the first file, and the pairs in the other. Okay, so I'll show you some of these outputs. Here's the PCR duplicate step. So, yep.
Can you get rid of the 1 minute more process in that case? And it wouldn't harm it.
Yes.
Does it necessarily matter any order?
It doesn't matter. I haven't actually tested it. I expect it would take longer if you did this first, because back here you have all individuals together. And this does, I don't think it does an all by all comparison, but it does some version of that. So once you have it split up by individual, you do that. There's also, so there are lots of different PCR duplicate removal programs out there. This one, like some others, depends on sequence similarity. There are also ones that depend on alignment position. So these are some part of SAM tools, I think, where basically you would skip the step here, go down, align all your repairs, and then run it through PCR duplicate removal that removes things based on whether or not they have identical alignment positions for the forward and reverse reads of any 2 pairs. Yeah, so there are different options for removing PCR duplicates, but again, they all depend on paired entities. Actually, I was just talking to Gordon this morning. We were talking about rates of PCR duplicates. In my lab, we found rates of PCR duplicates anywhere from 5% to 50% or so. Again, there are lots of things that can affect it, like primarily starting amount and quality of DNA, the number of samples that you're multiplexing. So the better the DNA you start out with, the more samples you multiplex into one library, the lower your rate of PCR duplicates will be. Okay, so here you can follow this reference aligned path, or you can follow this de novo path. Like I said, this way you go to U-stacks, which just essentially piles up reads into stacks depending on sequence similarity. Here you align to a reference, which stacks doesn't do internally. You have to use something like DWA, and then bring it back into stacks as SAM files, P stacks. And then these sort of paths merge again, you make a catalog, you match against the catalog. The output from that is what also you have some examples of. And then, like I said, you can output either population genetic statistics, or you can just output genotypes in all sorts of different formats for other programs. Okay, so the other things I think I mentioned in your handout, there's these sort of wrapper scripts. So there's one called RefMap, in other words, a reference map, which basically takes you from here, from SAM files, all the way through here, and maybe out that way if you specify the options. The de novo map takes you from here all the way down and maybe out that way if you specify those options. It's kind of your preference if you want to use those or if you want to do step-by-step. If you do step-by-step, you can see what's going on and maybe control some of the things a little better. But a lot of people like to use those wrapper programs because they press fewer buttons. And there are, on the website, which I'll show you in just a second, there are, like I said, there's a great user group, there's also tutorials and stuff, and all of these programs, you can look at all of the options to build the commands for those programs. So the data that I put on there is from Stickleback, from some Alaskan lakes. So they're in the data set, there's Arrow Lake and Tern Lake. That's Arrow Lake. That's Tern Lake, with Andrew McCall from Nottingham University. Pulling Stickleback out of the lake there. So, you have the partial set of data. What I would encourage you to do is just sort of play around with these different parts of stacks. Like I showed you on that little workflow, you can start from any point and go down from there. And try out the next step. Things like P stacks and U stacks will work per individual. So if you just have the one individual FASTQ file, you can run U stacks on the FASTQ or P stacks on the SAM file. If none of you have the full tarball, none of you will be able to do things like calculate FST between the different populations.
So that's the complete, or that's the small version?
This is the complete one
Yeah.
on the U of Idaho cluster. So just as an example, for if any of you have not looked at RAD data before, running this. So this is raw data right off the machine. Like I said, this is using the new protocol where the cut site can be on either end. So this is the first file. Yep.
Sorry, real quick, can you show us how to extract that tarball and use command line?
Oh yeah, right there.
Ah,
There are a bunch of options, Z, X, B, F. You don't have to have all of those. Hopefully that works on the doctor thing. So this is RAD data right off the machine. So this is the R1 file. So this is FASTQ format, 4 lines per read. If you look right here, there's TGC AGG. So this is a good red read. Those actually, it's not such a good red read. That is the barcode, which has an N in it, so it'll probably get thrown out at some point. But the way this protocol works, it always gives you these first two little nucleotides, which are usually GG. Those get trimmed off by that flip script that I put on there. Then you have the barcode. Then you have TGCAGG. So what enzyme did we use? SBF1 classic. So that's the TGCAGG. And then the rest of that is just random genetic sequence. But if you notice, so this first read, that's the cut site there. This second read doesn't have anything resembling that.
Mhm.
So hopefully, if it's a good read, which I don't think it actually is, but if it were, if you looked in the R2 file, the second read of the R2 file would have a correct barcode and TGCAGG. And then that pair would just need to be flipped. After processed rad tags, now it's been demultiplexed. The barcode has been trimmed off, and now you can see, and these have been all flipped also, now you can see they all start with TGCAGG. So this is the output of processed rad tags. And this would be ready to either align against the reference or go into USTATs to identify those cells. Any other questions?
You have to flip them first separately, but then the process rad tags cuts off everything for the tag sequence.
Right. And so,
And the flip is not part of stacks, that's some other script
Right.
you guys have.
right. So I think, Tiago, is there a flip script in the, you guys put one on Galaxy, right?
It's the local